Welcome back to my channel, and today we are going to study biochemistry in a very rapid manner. So first of all, certain concepts in biochem. There are two types of pathways in biochemistry, the anabolic pathway and the catabolic pathway. Anabolic pathways, all of the anabolic pathways, they occur in cytoplasm. Okay, so what include the anabolic pathway is glycogenesis, HMP shunt, whatever synthesis is going on, okay, like glycogenesis, HMP shunt, fatty acid synthesis, cholesterol synthesis, triglyceride synthesis, all these is occurring within the cytoplasm. The next is the catabolic pathway. Catabolic pathways, they mostly occur in the mitochondria except two of them, which is glycolysis and glycogenolysis, which occur in the cytoplasm. Rest all occur in the mitochondria. Catabolism is basically breakdown. So in whatever pathways you're breaking down something like glycolysis. The other examples are link reaction, that is pyruvate dehydrogenase reaction, glycogenolysis, okay, breakdown of glycogen and glycolysis. This occurs in the cytoplasm. Rest all are in the mitochondria. Then beta oxidation of fatty acids. Also, the gluconeogenesis, though it might seem to be genesis, but it is actually a catabolic pathway. Okay, so you have to remember that. And this gluconeogenesis, along with urea cycle and heme synthesis, this is occurring both in the mitochondria and cytoplasm. That means some part is in the mitochondria and some part of it is in the cytoplasm. Okay, then ketone body synthesis and ketone body utilization and breakdown are both catabolic pathways. See, you may not always consider like gluconeogenesis, so it is getting generated, so it's an anabolic pathway. No, ketone body synthesis is an anabolic pathway. No, it is catabolic pathway because this occurs when the body is in fasting stage and you have to break down your own body products to form glucose or ketone bodies. So gluconeogenesis, ketone body synthesis, and even ketone body utilization or breakdown, all of these are mitochondrial catabolic pathways. Gluconeogenesis is both in cytoplasm and mitochondria. And what other two things are occurring in both? Urea cycle and heme synthesis. In gluconeogenesis, 6 ATPs are involved. In urea cycle, 4 ATPs are involved. Okay, okay. After this basic understanding, the same thing that I was talking about in fasting and starvation. In fasting and starvation, what happens is the adipose tissue, it releases free fatty acids. This fatty acids goes into the liver and undergoes beta oxidation of fatty acid, which is a catabolic pathway. After beta oxidation, acetyl CoA is released, and then it enters into three pathways. The first one, either it may just enter into TCA, combine with oxaloacetic acid, and then form that citric acid and everything. So it may enter the TCA, or this acetyl CoA may get converted into the ketone bodies. So this ketone body synthesis, mitochondrial catabolic pathway, can again occur, or this may activate the gluconeogenesis, the first step. We'll read all of them so it will be more clear. And then this is utilized. So this occurs in the fasting or starvation state. Now, why this is occurring? Because there's no glucose. So let's just see what happens in with what stages of body. See, there are three stages. You can be in a fed stage, in a fasting stage, and starvation is prolonged fasting. Okay, so fed stage, fasting stage, and starving stage. Brain cannot utilize fatty acids, okay, as fatty acids cannot cross the blood-brain barrier. So it has only two options left: the glucose and the ketone bodies. And all you know is brain is the very important organ. So our body tries and preserves glucose even in the fasting stage. So fed stage mein to it is using glucose. Fasting stage maybe body is providing it with glucose. But when it's a starvation condition, then ultimately brain has to rely on ketone bodies. For heart, it utilizes fatty acid even in fed stage. So heart likes fatty acids. Okay. So even in fed and fasting, both stages it is utilizing fatty acids. and in starvation you have no fatty acid, no glucose, so it will, brain and heart both will utilize ketone bodies. Liver may in fed stage, all these, for all these peripheral organs, liver, muscle, adipose and RBC. In fed stage you are using glucose. When the fasting stage is arising, liver, muscle and adipose, they are okay with using fatty acids. Liver, in case of starvation stage, uses amino acids, which is quite unique because no other person is using amino acids. Okay, liver is using amino acids. Muscle में क्या है, that fed stage में you give him glucose, he'll use glucose. Fasting stage में you give him fatty acid, it will use fatty acid. But starvation stage में you give it whatever, it will use it. You give it glucose तो होता ही नहीं है, ठीक है, तो you give it fatty acid, it will you take it. Give it ketone body, it will take it. Give it amino acid, it will take it. So muscle is like very nice. We should be like this, adaptive. ओके, देन एडिपोज टिश्यू फास्टिंग में, देखो फेड में तो ग्लूकोज ही कर रहा है, इन फास्टिंग इट इज़ यूटिलाइजिंग व्हाट, फैटी एसिड. वेरी गुड. इन स्टारवेशन अगेन द एडिपोज टिश्यू के पास व्हाट इज स्टोर्ड इन इट, फैट. सो व्हाट विल इट यूज, व्हाट इज स्टोर्ड विद इन इट, so it uses fatty acid. Muscle में you can remember, muscle is made up of proteins, it is made up of some amount of fat also, some ketones can be formed, so it utilizes all three. Adipose use fatty acid. Specific thing about RBCs is they cannot utilize anything apart from glucose. So it has no other option. उसका पास कुछ और option ही नहीं है. It has no mitochondria, no other organ to utilize any other substance. So it just utilizes glucose, where be it fed, fasting, starving. Okay. And another thing was asked in previous year questions, that fetal heart, what does it utilize? It utilizes glucose. In heart failure, what it heart is utilizing? It's utilizing glucose. Okay, cool. Now certain OSA zones I want you to know, because these were quite commonly asked earlier. Whenever we are talking about glucose, fructose or mannose, needle shaped or broomstick shaped uh, structures are formed. Whenever we are talking about galactose, rhombic shaped. Whenever we are talking about maltosa zones or maltose, it forms sunflower. Okay, so you can see in sunflower this M is created, na, maltose. Okay, but not mannose. Remember that mannose is needle, maltose is sunflower, and then ultimately we have the lactose, which creates this hedgehog, cotton ball, or powder puff OSA zones. Okay, now there's something known as xanthoprotic reactions. This xanthoprotic reaction is given by all the aromatic amino acids except phenylalanine. What are the aromatic amino acids? Phenylalanine, tyrosine, histidine, tryptophan. Histidine and tryptophan are heterocyclic. So, and for tyrosine is derived from phenylalanine only. But all the aromatic amino acids are giving this xanthoprotic reaction on reaction with HNO3, except phenylalanine. So remember that. Then we have this Benedict's test. We all have done and performed Benedict test in our MBBS biochemistry days. So it's just nothing big. You can see the Benedict's reagent changes its color according to the amount of reducing sugar present in it. So it can be blue, it can be then green +1, +2 would be yellow, means traces is present. Orange is +3, which means moderate, and brick red is +4. Okay, large amount. Now, apart from the question asked actually is ki, what apart from the reducing sugar gives Benedict's test positive? So, uric acid, ascorbic acid, glutathione, creatinine, homogenesic acid, salicylic acid, cephalosporins. So, all this is actually making it can give a Benedict's positive test. Okay, so uric acid, ascorbic acid, glutathione, creatinine, homogentisic acid, salicylic acid, cephalosporin. All these are giving it positive. One more question, मतलब it's majorly acid you can see. एक cephalosporin से एक अलग है और एक creatinine and glutathione are different. Glutathione is made up of three amino acids: glutamate, cysteine, and glycine. You have to remember this: glutathione, cysteine, and glycine. Okay. Next we have the Molish test. Molish test is basically test for carbohydrates. It is negative in glyceraldehyde, positive in glucose ribose. It is positive in carbohydrates who have 5 or more carbon. And what happens is a purple ring is formed. Purple ring is also formed in Rothras. Rothras test for ketone bodies. Okay, so this purple ring is formed only if there are 5 carbons or more. Next, you have the biored test. Biored test occurs in proteins which have uh, 2 or more peptide bonds. Okay, so 2 or more peptide bonds will give it a positive. What is positive? A purple color will appear in biored test. So it is given by tripeptides and all the proteins ahead of it. Like glutathione, it is made up of 3 amino acids: glycine, cysteine, and glutamate. Okay, so it will have 2 peptide bonds. So it cannot give it positive. Okay, because it has only 2 peptide bonds. But sorry, it can give positive because it has 2 or more. मतलब you have to have 2 or more. It can give positive. But amino acids will not give positive. Now, ninhydrin test. Ninhydrin test is a test for alpha amino acids. Alpha amino acids में uh, all the alpha amino acids, but not proline. Okay, not proline. And above we read all the aromatic acids except phenylalanine. So what it gives is a purple color. Okay, again purple color. It's now not proline. Ninhydrin test is for alpha amino acids. You just have to remember that proline will give a yellow color in this case. And just a small table where will get to know about the specialized group and a bit of small test for each of them. Like histidine has an imidazole ring, and this imidazole ring can be given by Pauli's test. Tryptophan has an indole ring. Phenylalanine has a benzene ring. Tyrosine is a phenol. Arginine is guanetidium, and proline is pyrrolidine ring. Apart from that, methionine and cysteine methionine has thioester linkage and cysteine has thioalcohol linkage. Uh, the imidazole ring is present in histidine, indole in tryptophan, benzene in phenylalanine, phenol in tyrosine, guanidinium in arginine, proline में pyrrolidine. ठीक है, just remember these. And if you want to remember the test, I would want you to remember the Hopkins-Coley reaction of tryptophan, which is due to the indole ring, the Sakaguchi test for guanidinium, and if you want, you can remember Millen's and Folin test for phenol and Pauli's test for histidine. Okay. Now moving ahead. Moving ahead, we'll talk about gags. That is very important in forming connective tissue structures. They are hydrophilic, so they engulf water and become sponge like. Okay, glycosaminoglycans. So first one is hyaluronic acid. The specific thing about hyaluronic acid that it has no sulfate. Okay, so which gag has no sulfate? Hyaluronic acid has no sulfate. Okay, And second thing about it is the longest. Where it is present? In synovial vitreous wound healing. You know that. One specific is cell migration. Okay. In morphogenesis and tumor cell migration, it is actually involved to some extent. Next is chondroitin sulfate. Chondroitin sulfate is of all, most abundant of all, and it is present in cartilage, bone, tendon, ligament. Chondroitin sulfate. Next we have the keratin sulfate. Keratin sulfate के बारे में the specific thing is that it has no uronic acid. It is present in cornea and cartilage. Cornea and cartilage. Remember that. ठीक है? Keratin sulfate. Next we have dermatin sulfate, which is present in the blood vessel, skin, blood vessel and valves have dermatin sulfate. Heparin is intracellular and anticoagulant mast cell in liver में मिलता है. And heparin sulfate is cell surface. It has role in cell adhesion, like retinal cell attachment in the basement membrane of the glomeruli. You know, heparin sulfate is involved. Next we have the glute receptors. So you need to remember that all the glute receptors are bidirectional. First thing. Next thing, glucose dependent is GLUT 4. Glucose dependent GLUT, sorry, not glucose. Insulin dependent is GLUT 4. Okay, insulin dependent is GLUT 4. Okay. अब देखो GLUT 1 is present in the major organ, in the brain. If a lady gets pregnant, its baby is important to give it to baby. Placenta is important, so brain, placenta, kidney, and RBCs. ठीक है, these are the important structures. And what is GLUT1 doing? Providing the basal glucose transport, ठीक है, basal level पे. Then we have GLUT2. GLUT2 जो होगा, it is present in the intestine, liver, pancreas, kidney also. It helps in absorption. Okay, then liver, pancreas, kidney. In kidney it helps in reabsorption. Pancreas में insulin वाला part and liver में glycogen बनने का part is all this GLUT2 mediated. Intestine में तो basically it is causing absorption, kidney में reabsorption, liver में glycogen बनना है and insulin निकलना है pancreas से. This is GLUT2 mediated. GLUT3 is again present in brain, placenta and kidney, and it is again responsible for the basal glucose transport. So GLUT1 and GLUT3 are kind of similar. GLUT1 is specifically present in RBCs, which is different. You need to remember that. GLUT4 is present in all the other parts: skeletal muscle, cardiac muscle, adipose tissue. Insulin stimulated glucose uptake after meals occurs due to GLUT4. And GLUT5 is present in small intestine and testes and sperms, which is mainly responsible for the fructose transport. Mucopolysaccharidosis are a group of disorders which are very, very important for you to learn. And I have made out specifically very short, simple ways in order to revise this, which is available on my channel. Just go through the glycogen storage diseases, go through the lysosomal storage disorders, okay? And this is also important. Rest all we'll cover in the coming videos. Mucopolysaccharidosis has, the child will have certain specific features like coarse facies, growth retardation, mental retardation, short height, hepatosplenomegaly, bullet shaped middle pharynx is specific. This would be kind of the appearance, and though in the picture it might seem different, but Hunter has no clouding, Hurler has clouding. It is type 1 and autosomal recessive. Hunter is type 2 and X-linked recessive, does not have corneal clouding. Lysosomal storage disorders may you have a lot to remember, but just remember the Gaucher's disease, which is due to beta glucocerebrosidase, There is no mental retardation. Hepatosplenomegaly present easy bruising, bone pain, pathological fractures because it is involved in the bone. But no mental retardation, you need to remember that. And Gaucher cell या clumpal tissue paper appearance is characteristic. There's one more disease, Wolman disease or cholesterol ester storage disease, which is acid in which acid lipase is absent. Okay, Wolman disease, cholesterol ester storage disease. In this, there is calcification of adrenal, watery green diarrhea, hepatosmegaly, and there is relentless vomiting, excessive vomiting. Cholesterol and triglycerides are raised. Acid lipase की deficiency की वजह से होती है. Okay. So next we have the glycolysis. Okay, so we're talking about the glycolysis. Glycolysis is also known as EMP pathway, so you must not get confused. Embden-Meyerhof pathway is same as glycolysis, and we already have told you that glycolysis is a cytoplasmic process. Why? Because it is an anabolic process. Sorry, a catabolic process, but still it occurs in cytoplasm. See, when you do not revise, you forget. So you have to keep revising biochemistry. I forgot it even. I'm sorry for that. So glycolysis is a catabolic pathway. Obviously, it's a catabolic pathway. We're breaking down the glucose, but the thing is that even though catabolic, it occurs in the cytoplasm. Okay, good. Now moving ahead next, glucose is converted into glucose 6 phosphate. This glucose 6 phosphate, this occurs by the help of enzyme hexokinase or glucokinase, and ATP is used here. One ATP is used here. Okay, abhi you just have to look at this pathway which I'm teaching. Rest we'll discuss the other important stuff about it. So, being a cytoplasmic pathway, glucose is broken down into glucose. Glucose is not broken down actually, but converted to glucose 6 phosphate by addition of an ATP and hexokinase or glucokinase enzyme. Next, this glucose 6 phosphate is converted into fructose 6 phosphate by the help of enzyme isomerase. This fructose 6 phosphate is converted into fructose 1,6 bisphosphate. This is converted into fructose 1,6 bisphosphate. Again, one an ATP is used, and the enzyme which is involved here is phosphofructokinase 1, which is actually the rate limiting step. Next we have this fructose 1,6 bisphosphate by the help of enzyme aldolase A is converted into dihydroacetone phosphate and glyceraldehyde 3 phosphate, which are again interchangeable by the help of enzyme isomerase. So abhi tak it was all 6 carbons. Now they've just broken down, and we have glyceraldehyde 3 phosphate, which is a 3 carbon compound, so we can get just 2 molecules of this 3 carbon compound, glyceraldehyde 3 phosphate. Now this glyceraldehyde 3 phosphate, now due to NAD conversion and formation of 2 NADH, okay, 2 inorganic phosphates are used here. So 2 inorganic phosphates are used here. And this occurs by the help of enzyme glyceraldehyde 3 phosphate dehydrogenase, and it is converted into 1,3 bisphosphoglycerate. Okay, 1,3 bisphosphoglycerate molecule. 1,3 bisphosphoglycerate molecule. Now this 1,3 bisphosphoglycerate के 2 molecules are then converted into 3 phosphoglycerate by the help of enzyme phosphoglycerate kinase. 2-ADP are converted into 2-ATP molecule, and this is known as the substrate level phosphorylation. Okay, ATP is formed at the substrate level as well. You don't have to go to that ETC and everything. Okay, now 1,3 bisphosphoglycerate is converted into 3PG, 3 phosphoglycerate by the help of enzyme phosphoglycerate kinase. and this 3-phosphoglycerate is then converted into 2-phosphoglycerate, and ultimately this 2-phosphoglycerate by the help of enzyme enolase is converted into phosphoenol pyruvate. Okay, phosphoenol pyruvate is further the last step. Pyruvate kinase say it is converted into pyruvate, and again here 2 ADP are used up and 2 ATP is formed. So again a substrate level phosphorylation. So substrate level phosphorylation is occurring at pyruvate kinase and phosphoglycerate kinase levels. Um, then this pyruvate molecule so formed is actually converted to lactate by the help of enzyme lactate dehydrogenase, and NADH is converted into NAD. Now I want you to just go through the basic energy kinetics. See what happened during this aerobic pathway was that we were using ATP at two places, that is hexokinase and phosphofructokinase 1. So two ATPs were used up. Then two inorganic phosphates were used up at the level of this glyceraldehyde 3 phosphate dehydrogenase to Na phosphate level phosphorylation, substrate level phosphorylation. No, not this is not called substrate level phosphorylation. This is known as inorganic phosphate ka use. So two inorganic phosphates were used up, and then what led to the two inorganic phosphates were used up, and one and two NADH were formed. Okay, two NADH were formed. Then at two places 2 ATP were formed, phosphoglycerate kinase and pyruvate kinase. So ultimately, and we know, see, one NADH forms 2.5 ATP. So 2 NADH will form 5 ATP. So 5, 6, 7, 8, 9. 9 ATP were in total formed, out of which 2 ATPs were used up in the initial two steps. So 9 is the total in aerobic and 7 is the net. This is when malate aspartate shunt is being used. When glycerophosphate shunt is being used, it's 7 and 5. I will tell you this in a bit. But if question arises that during glycolysis malate aspartate shuttle was used or shunt was used, so you'll write the simple one. Then it says during this glycolysis glycerophosphate shunt was used. Then glycerophosphate shunt में 2 ATP कम produce हो गए, बाकी everything same, so it gets total as 7 and -2 to be net, तो 2 5 हो जाते हैं. Anaerobic में you're only gaining total of 4 ATPs, out of which 2 are utilized also, so net gain is only 2 ATPs. Fair enough. Now, important thing I want to tell you, that there are 3 irreversible steps: hexokinase, phosphofructokinase 1 and pyruvate kinase. ठीक है, insulin activates all these, and insulin कैसे activate करता है? By dephosphorylation. ah cool. Phosphofructokinase is actually considered kind of rate limiting enzyme, so इसका inhibitors तुम्हें याद रखना important है. It's ACP का mnemonic है ये, A for ATP, citrate and protons. They inhibit phosphofructokinase 1. Now coming on to what are the inhibitors of glycolysis. Fluoride inhibits enolase. Iodoacetate inhibits glyceraldehyde 3-phosphate dehydrogenase, and arsenate actually bypasses the PG kinase. So phosphoglycerate kinase is bypassed by arginase, is inhibited, enolase inhibited by sodium fluoride, and pyruvate kinase is inhibited by glyceraldehyde 3-phosphate dehydrogenase. This enzyme is inhibited by iodoacetate. Very good, very simple. Now RBC में 90% is ATP production through the normal glycolysis वाला pathway. Only 10% is Rapport shunt, जिसमें one substrate level phosphorylation is absent. This occurs only in RBC, only in 10%. 0 ATP is generated. That is, 2 is produced and 2 is used up. And why then does this occur? Because this leads to production of 2,3-BPG. Very cool, very simple. Moving ahead, I want to tell you something more about it. There are diseases in which G6PD deficiency is there and pyruvate kinase deficiency is there. To differentiate, both will present as hemolytic anemias. G6PD is the most common enzyme deficiency, pyruvate kinase is the second most common. Hemolysis will be absent. So hemolysis will be present in both. Heinz body. Heinz bodies will be absent in pyruvate kinase, present in their characteristic of G6PD deficiency. So they'll be present there. Okay. So I think we've covered glycolysis really very nicely. This is actually the minimum that you need to remember about it. Forget the rest. वो वैसे भी याद नहीं रहता. Then we're talking about Cori cycle. This a few years back, this was kind of becoming a favorite topic because either Cahill or Cori was interchangeably asked in the questions. देखो, Cori cycle or simply glucose lactate cycle is a cycle which occurs in the exercising muscles. During exercise, in the muscle, glucose is converted into pyruvate to lactate. This lactate, picked up from the muscle, goes to the liver, is converted back into pyruvate, and pyruvate se wapas glucose. And this continues. Then Cahill cycle. Cahill cycle या glucose alanine cycle. Cahill cycle or the glucose alanine cycle में glucose is converted into pyruvate, but this pyruvate is then converted into alanine. This is a fasting muscle. Okay, fasting state mein I told you muscle utilizes whatever available like amino acids, ketone bodies, fatty acid, whatever. So abhi it is trying and break down. So glucose is converted into pyruvate, and pyruvate by ALT enzyme is then converted into alanine. This alanine goes to the liver, converted to pyruvate, then to glucose, and then again back. So here alanine is there in Cahill and Cori lactate is there. नाउ ग्लूकोकाइनेज एंड हेक्सोकाइनेज के बारे में अगर हम बात करें तो ग्लूकोकाइनेज इज एक्चुअली प्रेजेंट इन लिवर एंड बीटा सेल्स ऑफ पैनक्रियास एट ऑल द अदर प्लेसेस हेक्सोकाइनेज इज प्रेजेंट ग्लूकोकाइनेज हैज़ अ लो एफिनिटी दो इट्स नेम इज ग्लूको यू कैन रिमेंबर लाइक ग्लूकोकाइनेज हैज़ लो एफिनिटी हेक्सो Okay, so hexokinase has high Km, high affinity, so low Km. You just remember one thing, yeah, the other thing comes its own way. Hexokinase has high affinity, so low Km, and vice versa for glucokinase. Type 1 to 3 enzymes of hexokinase is hexokinase, but glucokinase is the fourth isoenzyme. Hexokinase is inhibited by glucose 6-phosphate, but this glucokinase is not. Glucose kinase इंसुलिन की इंक्रीज एक्टिविटी से इंक्रीज होता है हेक्सोकाइनेज इज नॉट इट मेंटेन्स द ब्लड ग्लूकोज लेवल ग्लूकोकाइनेज hexokinase maintains the intracellular fluid glucose levels. Fine. Now, I was talking about the link reaction being a catabolic pathway which occurs in the mitochondria. I just want to teach you a bit about what this link reaction is. It involves pyruvate dehydrogenase complex, and this is commonly asked. In fact, the 3 enzymes of pyruvate dehydrogenase complex are asked. Give me 1 second, I just want to write those. Okay, so the 3 enzymes which are involved in this complex are number 1 pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl. Okay, so there are 3 enzymes: pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dehydrogenase. What are the 5 coenzymes which are needed in this pathway? It's B1, B2, B3, B5, and lipoic acid. Okay, so it's B1, B2, B3, B5, and lipoic acid. So these are the 5 coenzymes needed. One NADH forms 2.5 ATP. One glucose forms 2 pyruvate. Due to the this link reaction, NADH is formed from 1 pyruvate. So 2 will form 2 NADH. Now then in 2.5 it will be 0.5 ATP. So pyruvate in hydrogenase moves inside 3 carbon pyruvate, and by the help of enzyme pyruvate dehydrogenase complex is converted into acetyl CoA. 3 carbon होता है pyruvate carboxylase, sorry pyruvate 3 carbon होता है, and pyruvate dehydrogenase complex से act करती है, तो NADH बनता है, और क्या बनता है, एसिटाइल कोए बनता है. ये देखो, दिस acetyl CoA, this acetyl CoA is formed, NADH is formed. Fine. NADH बन गया, acetyl CoA बन गया, and carbon dioxide is released. Okay, this is released, carbon dioxide. B1 deficiency में, this is the reason why lactic acidosis can occur, because this pathway will not occur, and pyruvate will get converted into lactic acid. Fine, this is what you have to remember. Moving ahead, now next we have the TCA cycle. I know this is quite boring, but we have to remember that. TCA cycle में सबसे पहले you start here. See, pyruvate was the product of glycolysis. Now this pyruvate is converted by the help of link reaction by the formation of NADH into acetyl CoA. This acetyl CoA, which is a 2 carbon compound, is then combines with the oxaloacetate 4 carbon compound to form a 6 carbon citrate by the help of enzyme citrate synthase. Then aconitase enzyme की वजह से it is converted into isocitrate. Then isocitrate dehydrogenase की मदद से it is converted into alpha ketoglutarate. Okay, and dehydrogenase में 1 NADH is formed. 1 NADH is formed. Okay. Then alpha ketoglutarate dehydrogenase is converted into succinyl CoA. This succinyl CoA, this conversion is by the help of enzyme alpha ketoglutarate dehydrogenase, again then NADH. Then succinyl CoA is then converted into succinate by succinate thiokinase enzyme. फिर succinate to fumarate by succinate dehydrogenase. Here FADH2 is formed. Okay, and this happens in the mitochondrial membrane. Then fumarate to malate by fumarase, and malate dehydrogenase converts into oxaloacetate. Again, 1 NADH is formed. At this step of succinyl CoA conversion to by the help of enzyme succinate thiokinase to succinate. Here an ADP is converted into an ATP in liver, kidney, fasting, starvation, GTP, as gluconeogenesis need GTP. Okay, now the energetics which is involved here is, we saw at 3 places, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and malate dehydrogenase, NADH were formed. So 3 NADH into 2.5, that is 7.5 ATP. Then at one place succinate dehydrogenase 1 FADH2 was formed, so it's 1.5 ATP, and 1 ATP was a substrate level phosphorylation at succinate thiokinase step. So total of 10 ATP is formed from 1 acetyl CoA, irrespective of whether it's aerobic, anaerobic, falana dhimakla, any kind of TCA will form 10 ATP. Okay, so from 1 acetyl CoA it's 10, from 2 acetyl CoA it will be 20 ATP. So 1 glucose will form 2 acetyl CoA, so 20 ATP from TCA. Okay, now there is something that I want to tell you, the complete energetics. So 1 glucose was forming 2 pyruvate, was forming 2 acetyl CoA, which were entering the TCA. So 1 glucose to 2 pyruvate was forming 7 ATP. Then 2 pyruvate to 2 acetyl CoA, एक NADH बन रहा था, अब 2 हो गए 5 ATP. एंड 2 एसिटाइल कोए से 20, सो टोटल ऑफ 32 ATP का गेन होता है, ओके, इन एन एनारोबिक साइकिल. अब मैं छोटा सा बता दूँ TCA के इनिबिटर्स. सो देयर आर सर्टेन स्टेप्स जिसमें हर कोई इनिबिट कर रहा है. फ्लोरोएसिटेट इज इनिबिटिंग द एकोनिडेज एंजाइम. Fluoroacetate is inhibiting the aconitase. Arsenite is inhibiting the alpha ketoglutarate dehydrogenase, and malonate is inhibiting succinyl dehydrogenase. Okay, so fluoroacetate is inhibiting this aconitase enzyme, is inhibited by fluoroacetate. This alpha ketoglutarate, I don't want to do it such big thing, want it to be small, so okay. Aconitase is inhibited by fluoroacetate. The arsenite is inhibiting alpha ketoglutarate dehydrogenase, and the malonate, this malonate is inhibiting the succinyl dehydrogenase enzyme. Okay, succinyl dehydrogenase, this is inhibited by malonate. So at 3 places we have 3 inhibitors. The fluoroacetate inhibiting the aconitase is non-competitive. The arsenite inhibiting the alpha ketoglutarate is non-competitive. The malonate inhibiting the succinyl dehydrogenase is competitive. Okay, let me just write it. This aconitase, this is inhibited by fluoroacetate. Then this alpha ketoglutarate, okay, this is inhibited by arsenite. Okay, and the last succinyl dehydrogenase, this is inhibited by malonate. This is competitive, the rest two are non-competitive inhibitors. And revising the inhibitors of glycolysis, so at 3 steps we had fluoride was inhibiting enolase, we all know that. Remember this glucose 3 phosphate dehydrogenase by iodoacetate and bypass the PG kinase by arsenate. Okay, this bypass is by arsenate. And iodoacetate is inhibiting glucose 3 phosphate dehydrogenase. Glyceraldehyde 3 phosphate dehydrogenase. This is inhibited by iodoacetate. ठीक है, आयोडोएसिटेट यहाँ हो गया, फ्लोरोएसिटेट यहाँ हो गया, आर्सेनाइट भी यहाँ हो गया, मैलोनेट भी यहाँ पर ही हो गया. So I hope I am clear with all these thing. arsenite वहाँ है, arsenate यहाँ है, फ्लोरोएसिटेट वहाँ है, आयोडोएसिटेट यहाँ पे है. Okay, so that's it. Enolase तो we already know, sodium fluoride. That's it. Malonate का याद रखना. Now we are moving on to another very important part of biochemistry, which is the electron transport chain. This electron transport chain is made up of 5 complexes, actually 4 major, which are arranged in an increasing order. These are arranged in an increasing order of redox. Okay, increasing order of redox potential. Now this complex 1, I will try to show it to you here at the end. Abhi you just read that complex 1 is NADH coenzyme Q reductase. Okay, electron transport chain. The complex 1 is NADH coenzyme Q reductase, जिसमें 4 hydrogen ions का movement होता है at the NADH FMN point. The complex 2 is succinate coenzyme Q reductase, जहाँ पे FAD is involved. Third complex is coenzyme Q cytosine 3 reductase, and CYTC oxidase is complex 4. ठीक है. तो कॉम्प्लेक्स 3 क्या है? कोएंजाइम क्यू साइटोक्रोम सी रिडक्टेज, जिसमें अगेन 4 एच आयन का गेन है। इसमें बी और C1 साइटोक्रोम इन्वॉल्व होता है। और फोर्थ वाले में क्या है? साइटोक्रोम सी ऑक्सीडेज, जिसमें सिर्फ 2 एच आयन इन्वॉल्व है। एंड साइटोक्रोम A3 इज द वन। ठीक है, तो अब और कॉम्प्लेक्स 5 क्या होता है? एटीपी प्रोड्यूसिंग वाला जो फाइनल होता है, जिसमें एफ नॉट एंड F1 सब यूनिट्स होते हैं ना, ये F नॉट और F1, ये जो होता है ये हमारा कॉम्प्लेक्स 5 है, जहाँ पे एक्चुअल एटीपी प्रोडक्शन हो रही है। ठीक है, तो यू जस्ट हैव टू गो थ्रू दिस। देखो कॉम्प्लेक्स 1 क्या था? NADFMN था। NADFMN का क्या हुआ इसमें? NAD से NADH से NAD बना और 4H+ ने मूव करा। फिर दूसरा वाला क्या में क्या हुआ? सक्सिनेट कोएंजाइम क्यू रिडक्टेज वाला था ना, तो सेकंड वाले में कोएंजाइम क्यू बीइंग इनवर्ड FAD की वजह से नथिंग इज अकरिंग एट कॉम्प्लेक्स 2। कॉम्प्लेक्स 3 में कोएंजाइम क्यू साइटोक्रोम सी, ओके, BC1 वाला, इसमें 4H+ आर मूविंग। एंड देन लास्ट कॉम्प्लेक्स 4 इज साइटोक्रोम सी ऑक्सीडेज। दिस साइटोक्रोम सी ऑक्सीडेज, साइटोक्रोम सी ऑक्सीडेज की उससे A3 आर द कॉम्प्लेक्स साइटोक्रोम है, जिसमें सिर्फ टू टू मूव करेंगी, टू। ठीक है, नाउ हाउ मैनी H+ आर मूविंग इन? 4 4 8 9 10। ठीक है, तो 4H+ आर फॉर्मिंग 1 ATP। यहाँ से 4H+ अंदर जाएंगे तो 1 ATP प्रोड्यूस होगा। तो अगर मेरे पास 10 है तो कितना होगा? 2.5 ATP प्रोड्यूस। ठीक है, तो 2.5 ATP इज़ प्रोड्यूस्ड एक बार में। इन सब की वजह से एक बार अगर काम करते तो 2.5 ATP इज़ फॉर्म्ड। 2.5 ATP is formed. Now the inhibitors of ETC, very important. So inhibitors of everything is very important. So electron transport chain में you have rotenone, phenobarbital, pyrisidine and amobarbital, जिसका mnemonic था PAR। तो कॉम्प्लेक्स 1 को कौन इनिबिट कर रहा है? PAR। phenobarbital, pyrisidine, amobarbital, rotenone। 2 वाले का इनिबिटर होता है मैलोनेट, जो सक्सिनेट डीहाइड्रोजनेज का भी था। 3 वाले का एंटीमाइसिन सी बाल और फेनफॉर्मिन होता है। एंड 4th वाले का कार्बन मोनोऑक्साइड, साइनाइड, सोडियम अजाइड, हाइड्रोजन सल्फाइड। 2 other things, etractyloside inhibits ADP ATP antipode, which is actually inhibiting the ATP transfer. Oligomycin is inhibiting the F naught channel, so it is actually inhibiting the ATP conversion. And then certain uncouplers, which can be natural like bilirubin, thermogenin, T4, or they can be chemical like 2,4-dinitrophenol, 2,4-dinitrocresol. Very fair. Okay, gluconeogenesis. Gluconeogenesis is occurring, I told you there are 3 pathways: gluconeogenesis, urea cycle, and the heme synthesis. They occur both in the cytoplasm and the mitochondria. So though it is a catabolic pathway, gluconeogenesis, it is occurring in both, and cyto and mito. कैसे बनता है? I have to generate glucose. So I have, how am I going to do this? I'm going to reverse the step of glycolysis, obviously. But what all substrate can I use to make glucose? I can have pyruvate, I can have lactate, I can have glycerol, I can have propionic acid, I can have glucogenic amino acids, and I can have both ketogenic glucogenic amino acids. So all these are important and you have to remember, but I will try and remember the ultra important stuff. Okay, so we're starting it. Let's just start up from here, that is pyruvate. Let's imagine that you have pyruvate here. Pyruvate, by the help of enzyme pyruvate carboxylase, is converted into oxaloacetate. This oxaloacetate is converted into malate, and then this malate moves out of the mitochondria. Okay, moves out of the mitochondria. And goes out. Again, malate is converted back to oxaloacetate, and oxaloacetate by the help of enzyme phosphoenol pyruvate kinase is converted directly into phosphoenol pyruvate. Now this phosphoenol pyruvate is returned back to fructose 1,6 bisphosphate. All are reversible steps. Then an irreversible steps come, and now you have to use the enzyme fructose 1,6 bisphosphatase. Now it converts it into fructose 6 phosphate. Then again isomerase to use होगा, glucose 6 phosphate बनेगा, and glucose 6 phosphatase से glucose ultimately you'll be able to form. तो glycolysis में there were 3 irreversible enzymes: hexokinase, phosphofructokinase 1 and pyruvate kinase. Pyruvate kinase को you bypass by pyruvate carboxylase enzyme or Pepsi K enzyme. Phosphofructokinase 1 को you bypass by fructose 1,6 bisphosphatase, and hexokinase को you bypass by glucose 6 phosphatase. Fair enough? You've got it? Okay, move ahead. Another very important one is this: both glycogenesis and glycogenolysis together. And glycogenesis is a what? Glycogenesis is you're forming glycogen, and glycogenolysis is you are breaking it down. Glycogenesis and glycogenolysis. This glycogenolysis is actually a cytoplasmic step, even though it is catabolic pathway. I told you, even though catabolic, glycolysis and glycogenolysis, and also gluconeogenesis, urea and heme to chalo chhod do, they are occurring both, they are occurring in cytoplasm. Gluconeogenesis both in cytomito. So glycolysis, glycogenolysis and glycogenesis, अभी हम इसके बारे में बात कर रहे हैं, ठीक है. ग्लाइकोजन के दो पाथवे, ग्लाइकोजन बनना और ग्लाइकोजन टूटना. ग्लाइकोजन बनना इज ग्लाइकोजेनेसिस, एनाबॉलिक पाथवे साइटोप्लाज्म में ही होना है. कैटाबॉलिक पाथवे ग्लाइकोजिनोलाइसिस, मतलब ग्लाइकोजन का टूटना कैटाबॉलिक है, मगर इट शुड हैव अकर्ड इन द माइटोकॉन्ड्रिया, बट स्टिल इट इज अकरिंग इन द साइटोप्लाज्म. Similar cases with glycolysis, and somewhat similar is also gluconeogenesis. So basically what I want to tell you is this is occurring in the cytoplasm. बस मैं इतना कहना चाह रही हूँ. I am just getting confused a bit. No problem. When you revise, read it daily, you will be thorough with it. So there's no need to worry. ठीक है. Glycogenesis and glycogenolysis both are occurring in the cytoplasm. पहले हम लोग glycogenesis की बात कर रहे थे. How are we forming glycogen? Glucose होता है. Glucose by the help of enzyme glucokinase, hexokinase, glucose 6 phosphate में convert होता है, and then by the action of enzyme mutase, it is converted into glucose 1 phosphate. This glucose 1 phosphate by the help of enzyme UDP glucose phosphorylase and UTP is used, it forms UDP glucose. This UDP glucose से ग्लाइकोजन सिंथेज, जो कि एक रेट लिमिटिंग एंजाइम है, इसमें ग्लाइकोजन सिंथेज एक्ट करता है. 1,4 ग्लूकोज लिंकेज होती रहती है, लंबी लंबी चेन्स बन जाती हैं. देन द ब्रांचिंग एंजाइम एक्ट्स and then it forms the glycogen proper. Glycogen storage disorders kabhi I have a separate video, go watch it and you would be able to remember them quite easily. Like in branching enzyme deficiency, the disease is known as Anderson disease. Okay, because it's the mnemonic. I've told you the mnemonic also there, A B C D, A के बाद B आता है, C के बाद D आता है. तो एंडरसन में ब्रांचिंग एंजाइम डेफिशिएंसी है, कोरे में डी ब्रांचिंग एंजाइम डेफिशिएंसी है. We'll read it, don't worry. Now अब ग्लाइकोजन बन गया है. Now ग्लाइकोजन माइनर ग्लाइकोजिनोलाइसिस होगा तो एसिड माल्टेज काम करता है एट अ माइनर लेवल. लेकिन मेजर लेवल का जो ग्लाइकोजिनोलाइसिस है ना, उसके लिए यू नीड दीज 3 एंजाइम: ग्लाइकोजन फॉस्फोराइलेज विच इज़ द रेट लिमिटिंग एंजाइम हियर एंड नीड्स व्हाट, B6 ग्लूकन ट्रांसफेरेज एंड डी ब्रांचिंग एंजाइम. ग्लाइकोजन फॉस्फोराइलेज डेफिशिएंसी अगर मसल में होती है, इट लीड्स टू मैकार्डल्स डिजीज, लिवर में होती है, इट लीड्स टू हर्स डिजीज। डी ब्रांचिंग एंजाइम की डेफिशिएंसी से कोरी या फोर्ब्स डिजीज या लिमिट डेक्सट्रिनोसिस हो जाता है, बिकॉज इट लीड्स टू अक्यूमुलेशन ऑफ लिमिट डेक्सट्रिन्स। एसिड माल्टेज डेफिशिएंसी लीड्स टू पॉम्पेज डिजीज। वो फिजिक्स केमिस्ट्री और मैथ्स हेट करता था, so आई हैव टोल्ड यू निमोनिक्स इन दैट वीडियो, गो जस्ट वॉच इट, ओके। वो से वॉन गिल्कीज डिजीज जिसमें ग्लूकोज 6 फॉस्फेटेज। ओके, आई वाज जस्ट टीचिंग यू ना। हाँ, ग्लाइकोजन बाय द हेल्प ऑफ दीज एंजाइम्स ग्लाइकोजन फॉस्फोराइलेज ग्लूकन ट्रांसफेरेज 3 ब्रांचिंग एंजाइम is converted into glucose 1 phosphate. Glucose 1 phosphate is then converted into glucose 6 phosphate, and by glucose 6 phosphatase it is converted back into glucose. Von Gilkey's disease is the disorder in which glucose 6 phosphatase is deficient, and Pompey's disease is acid maltase deficiency जिसमें minor glycogenolysis is involved. So this is what I was talking about. वो फिजिक्स केमिस्ट्री और मैथ्स हेट करता था। सो वो वॉन गिल्कीज मोस्ट कॉमन ग्लाइकोजन स्टोरेज डिसऑर्डर, डॉल लाइक फेसिस, हेपाटोस्प्लिनोमेगली, रीनोमेगली, हाइपर यूरिक एसिड, लिपिड एंड कीटोन, एंड बट देर इज हाइपोग्लाइसीमिया। ओके, बट देर इज हाइपोग्लाइसीमिया। ये सब हाइपर है, ठीक है, बट देर इज वॉट इज नोन एज hypoglycemia because you are not able to do this glycogenolysis. Enzyme glucose 6 phosphatase is deficient, enzyme liver is involved. Organ liver is involved. Type 2 is Pompey's acid maltase deficiency, liver, muscle, brain, heart all is involved. हार्ट में पंपिंग डिफेक्ट होता है पॉम्पी में। कोरी लिमिट डेक्सट्रिनोसिस, डी ब्रांचिंग एंजाइम डेफिशिएंसी, all the three: liver muscle, heart, liver muscle, brain. Anderson again, same branching deficiency. McArdle's most common in adult, most common in children, von Gerke. McArdle's में lactate is normal after exercise. Muscle glycogen phosphorylase deficiency, and hers में muscle phosphorylase deficiency, liver glycogen phosphorylase deficiency. Okay, very good. Now I talk to you that this glycogen phosphorylase is the rate limiting step in glycogenolysis. So this glycogenolysis का step, glycogen phosphorylase. This is important to be read कि इसके एक्टिवेटर्स कौन है, इसका इनहिबिटर्स कौन है. Now this glycogen phosphorylase, when will it be active? In fasting stage, यानी कि when insulin is less, so it is activated in phosphorylated state because insulin causes dephosphorylation. It is active in phosphorylated state, insulin deficient state, fasting state. एक्टिवेटर्स कौन कौन है? वट ऑल विट वॉन्ट कि ये एक्टिव हो एंड ग्लूकोज बने, ग्लाइकोजन टूटे, तो साइक्लिक ए एम पी, ग्लूकागन, एपिनेफ्रिन, नॉरएपिनेफ्रिन, कैल्शियम एंड 5-AMP. बट वॉट विल से नहीं वी हैव इनफ ऑफ ग्लूकोज, यू स्टॉप ग्लूकोज इट सेल्फ, ग्लूकोज, इंसुलिन, ग्लूकोज 6 फॉस्फेट, ATP, ठीक है. अपार्ट फ्रॉम दैट 2 more, प्रोटीन फॉस्फेटेज एंड फ्रक्टोज 1 phosphate will actually tell you this. Cool. So we are done with this also. Now we are going to begin another few small topics. Before that, I told you that glycogenolysis, gluconeogenesis. Gluconeogenesis mein you can use certain ketogenic and glucogenic amino acids, or simply who are plainly glucogenic amino acid. So amino acids can be ketogenic, glucogenic, or they can be both. Okay, now we'll talk about the amino acids. See, amino acids, you just have to remember two things. There are ketogenic amino acid, leucine and lysine, who cannot make glucose. And there are amino acids which can produce both glucose and ketones. This is tip is the mnemonic. A tip, तुमने 5 रुपए की tip दी. Okay, when you go to restaurants you give tip na, so you gave 5 rupees tip. T for tyrosine, tryptophan, threonine, isoleucine and phenylalanine. Okay, I think we can remember it. Tyrosine, tryptophan, threonine, isoleucine and phenylalanine. Glucogenic mein most glucogenic is alanine and rest all are glucogenic only. Fats cannot be converted into carbohydrates except glycerol, जो बन सकता है ट्राइग्लिसराइड से, अ नॉट चेन फैटी एसिड हु कैन फॉर्म प्रोपियोनिक एसिड. सो फैट्स में सिर्फ यही दो फैट के बाई प्रोडक्ट हैं हु कैन मेक इट. ओके, so I think that's all for today. Tomorrow we'll do galactose and a bit of general part and few diseases. Till then, goodbye.